In this video, we are going to talk about nitric oxide, a signaling molecule that plays a prominent role in vasodilation. Nitric oxide was first identified as a gas by Joseph Priestley in 1772, where they found it is a simple molecule consisting of just one atom of nitrogen and one atom of oxygen, so is also called nitrogen monoxide. But for much time, it was thought of as an atmospheric pollutant only. But later in the 1980s, researchers started investigating vasodilatation inside the body, which is extremely important for blood pressure control. At that time, nitroglycerin was given to patients with heart conditions in order to promote vasodilation, but their mechanism was not known then. Later in 1980s, Robert Furchtgott investigated the role of a drug called acetylcholine on vasodilation, and they found that relaxation of blood vessels only occurred if a special class of cells called endothelial cells were present. Behind the endothelial cells is another specialized type of cell known as smooth muscle cells, and it was thought that the contraction and relaxation of these muscle cells are thought to be responsible for constricting or dilating the blood vessels. But then Robert Furchtgott and his group found that without endothelial cells, these smooth muscles were not able to cause vasodilation alone. So they suggested that there was some kind of factor produced by the endothelial cells that were required for the relaxation of blood vessels, and they named it endothelial-derived relaxing factor, EDRF. Independently, in 1977, Ferid Murad was investigating how nitroglycerin works and discovered that it can release nitric oxide, which in turn was able to cause the relaxation of smooth muscle cells. And then finally, in 1986, Luis Ignarro identified EDRF and found it has identical properties to the gas nitric oxide. They were awarded the Nobel Prize for this valuable discovery. Let's have a look at its biosynthesis now. Arginine, which is an amino acid, and other nitrates that are obtained through diet or are endogenously produced inside the body, are the main source of production of nitric oxide in the human body. Here, a family of nitric oxide syntheses are the enzymes that play a role in the production of nitric oxide from these substances. And these NOS are present in many cell types, including vascular endothelial cells, macrophages, neurons, and renal tubular cells, but mainly these are found in kidneys. Nitric oxide syntheses are classified into two categories: endothelial NOS and neural NOS, where endothelial NOS are further two types: constitutive NOS, also known as type 3, and inducible NOS, also known as type 2. This endothelial NOS are those that are derived from endothelial cells, and here the constitutive NOS are present constitutively in the kidneys, whereas inducible NOS are expressed after transcriptional induction and remains active for prolonged periods. And neural NOS are those that serve as a transmitter in the brain and in different nerves of the peripheral nervous system. Now here, all these three isoforms of nitric oxide synthase play a role in the production of nitric oxide. And this production of nitric oxide is calcium dependent in the case of constitutive and neural nitric oxide synthase, and is calcium independent in case of inducible nitric oxide synthase. Let's see how. The activity of constitutive nitric oxide synthase is calcium, and also calmodulin dependent. Here, calmodulin is an intermediary protein that senses calcium levels. Then there are two pathways of constitutive NOS activation. Both require calcium ions. The first one is the flow-dependent nitric oxide formation. And another one is the receptor-stimulated nitric oxide formation. In flow-dependent nitric oxide formation, shearing forces acting on the vascular endothelium generated by blood flow causes a release of calcium and thus subsequent constitutive nitric oxide synthase activation. While in receptor-dependent nitric oxide formation, endothelial receptors activate a variety of ligands that stimulates calcium release and thus ultimately causes constitutive NOS stimulation. On the other hand, the inducible NOS activation is calcium independent. However, its activity, like all of the NOS isoforms, is dependent on the binding of calmodulin. Increases in cellular calcium leads to increases in levels of calmodulin, and the increased binding of calmodulin to endothelial and neural NOS leads to a transient increase in nitric oxide production by these enzymes. By contrast, inducible NOS is able to bind tightly to calmodulin even at a very low cellular concentrations of calcium. Consequently, inducible NOS activity doesn't respond to changes in calcium levels in the cell. Under normal, basal conditions, the activity of inducible NOS is very low. The activity of inducible NOS is stimulated during inflammation by bacterial endotoxins, for example, lipopolysaccharide, and cytokines such as TNF and interleukins. During inflammation, the amount of nitric oxide produced by inducible NOS may be a 1,000-fold greater than that produced by constitutive NOS. In this way, during bacterial or other infection, inducible NOS gets activated. So, once all or any of these NOS gets activated by either of the mechanism described, then these nitric oxide syntheses catalyze the oxidation of L-arginine to nitric oxide and L-citrulline by utilizing NADPH and an oxygen molecule. This nitric oxide thus produced play a very important role in the vasodilatation of blood vessels. When nil forms, it has a half-life of a few seconds, as it has a very high affinity for superoxides. Once released by the endothelium, nitric oxide diffuses into adjacent and downstream vascular smooth muscle cells. Nitric oxide thus avidly binds to the heme moiety of the enzyme guanylyl cyclase, which is found in vascular smooth muscle cells and most other cells of the body. This enzyme catalyzes the dephosphorylation of GTP to cGMP, which serves as a second messenger for many important cellular functions, particularly for signaling smooth muscle relaxation. This cyclic GMP induces smooth muscle relaxation by multiple mechanisms including increased intracellular cGMP, which inhibits calcium entry into the cell and decreases intracellular calcium concentrations. Also, it activates potassium channels, which leads to hyperpolarization and thus relaxation. Also, it stimulates a cyclic GMP-dependent protein kinase that activates myosin light chain phosphatase, the enzyme that dephosphorylates myosin light chains, which leads to smooth muscle relaxation. So, by all these mechanisms, cGMP causes vasodilation. In this way, nitric oxide play a role in vasodilation and thus the relaxation of smooth muscles. Since nitric oxide was first discovered to play a role in the dilation of blood vessels only. But now many new roles for nitric oxide have been discovered. Nitric oxide has been found to be produced by virtually every cell type in the body and plays an important role in controlling the normal function of cells, as well as in regulating larger-scale processes such as the nervous and immune systems. Like in the immune system, nitric oxide plays an important role. where it is produced in high amounts from macrophages, following the activation by cytokines in a bacterial infection. The high amounts of nitric oxide produced by the macrophages are actually toxic to the bacteria and play an important role in their destruction. However, too much nitric oxide production has also been implicated in conditions where the immune system is too active, diseases like arthritis and the so-called autoimmune diseases. Then in the nervous system. Nitric oxide has been shown to be involved in both the central and peripheral nervous systems. We know that neural NOS are found in the nervous system, where they produce nitric oxide, and this nitric oxide then serves the role to transfer nerve signals from one neuron to another. It basically stimulates the production of neurotransmitters that are released from neurons at the synaptic cleft. In this way, it plays a vital role in signal transmission. Also, nitric oxide is known for its role in reproduction. Nitric oxide is involved in many aspects of reproduction. It is thought to play a role in the implantation of the early embryo in the uterus, and it functions to relax blood vessels, and thereby helps to regulate maternal blood pressure. During pregnancy, nitric oxide may also play a role in promoting the formation of new blood vessels, a process known as angiogenesis. It is also known to be an important survival factor for specialist cells called trophoblasts, which form the placenta. Other than these roles, a wide range of cellular activity is also regulated by nitric oxide, including cell division, cell survival, and cell movement. Most cells have an inbuilt self-destruct system or cell suicide mechanism. This mechanism, usually called apoptosis or programmed cell death, exists to prevent damaged or infected cells from affecting the proper functioning of the rest of the tissue. Nitric oxide has been shown to inhibit apoptosis and therefore is important in promoting cell survival. However, high doses of nitric oxide have been reported as being toxic to many cell types, and in these circumstances may promote cell death instead. So, this was all about the introduction of nitric oxide, its biosynthesis, mechanism of action, and physiological importance. For more such interesting videos, keep visiting sqadia.com.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.